Life cycle of ameloblast. Ameloblasts are cells that secrete enamel and they present only during the tooth development. Amelogenesis. It is the process of formation of enamel by ameloblast and it occurs in two stages, that is organic matrix formation and mineralization. Mineralization is again divided into phase 1 and phase 2. Phase 1 mineralization, 25 to 30% mineralization takes place. In phase 2 mineralization, 70 to 75% mineralization takes place. Enamel, dentine, cementum, bone all has got inorganic part and organic part. So for enamel, 96% is inorganic and 4% is organic. Coming to life cycle of ameloblast. Based on function of inner enamel epithelial cell, the life cycle of ameloblast is divided into 6 stages. That is morphogenic, organizing, formative, maturative, protective and desmolytic. These 6 stages can again be divided into 3 categories. They are pre-secretory, secretory and post-secretory. Pre-secretory stage include morphogenic and organizing stage. In this stage, the inner enamel epithelial cell prepare to secrete enamel. Secretory stage include formative stage and here the actual enamel formation takes place. Post-secretory stage include maturative, protective and desmolytic stage and this stage include the maturation and protection of enamel. As already mentioned, amelogenesis that occurs in 2 stages, that is organic matrix formation and mineralization. Here organic matrix formation that takes place in formative stage and phase 1 mineralization that takes place in formative and phase 2 mineralization that takes place in maturative stage. So let's discuss each stage in detail. So first stage, it is the morphogenic stage. Inner enamel epithelium determines the morphology of the tooth, hence the name morphogenic stage. It occurs in early bell stage. So here you can see the early bell stage. So there is outer enamel epithelium, stellate reticulum, stratum intermedium, inner enamel epithelium, dental papilla and there is dental sac. So this portion is just enlarged here. So here we have outer enamel epithelium, stellate reticulum, stratum intermedium, inner enamel epithelial cell, basement membrane and dental papilla. Here there is a zone, cell-free zone of dental papilla. Then the cell has got a proximal end and a distal end. Proximal end, it is the side which is closer to the stratum intermedium. Distal end which is closer to the dental papilla. So let's see the changes that taking place in the inner enamel epithelial cell. So in morphogenic stage, the cell is low cuboidal or short columnar cell with a large nucleus and mitochondria is evenly distributed in the cytoplasm. But the centrioles and Golgi bodies are located in the proximal end. And the cell rests on the basement membrane and it is separated from dental papilla by a cell-free zone. Next is organizing stage. This stage occurs in late bell stage and in this stage the inner enamel epithelial cell organized to form the ameloblast, hence the name organizing stage. In this stage the cell is elongated up to 40 microns and it becomes tall columnar. Also the nucleus moves to proximal end and that create nucleus free zone and the distal end and this is called reversal of polarity. And also the mitochondria move to proximal end. The Golgi bodies and centriole move to the distal end. And here also you can see a basement membrane. Here the cell-free zone of dental papilla disappears. So ameloblast comes in contact with the dental papilla and it will induce the formation of odontoblast and this odontoblast secrete dentine in this organizing stage. Next is formative stage. This is the stage in which actual enamel formation takes place, and this occurs in advanced Bell stage. Here you can see the ameloblast has increased in its length, and also it moves away from the dentine and form a pyramidal process known as Tom's process. And Tom's process is separated from cell proper by incomplete septa known as distal terminal bars. And here you can see there is no basement membrane. And coming to the nutrition, in the initial stages, it was from dental papilla. But after hard tissue formation, the nutrition is transiently from the stellate reticulum and then from the dental sac. Let's discuss Tom's process in detail. Tom's process has got two parts, a non-secretory side and a secretory side. Secretory side is again divided into proximal and distal. The proximal portion that secrete interrod, and the distal portion secrete rods. Always interrod formation is ahead of the rod formation. Here you can see the section of enamel prism. In the transverse section, it has a keyhole appearance with head and tail. But in longitudinal section, they occur as rods and interrods. That means the head portion corresponds to rods and the tail portion corresponds to interrod. Coming to maturative stage, this stage is 3 times longer than that of the formative stage, and mainly two processes takes place. That is, organic matter, it is reabsorbed from enamel, and the minerals are released onto the enamel. 50% of the ameloblast undergo apoptosis. Here, the basement membrane reappears. Then you can see ameloblast with smooth border and ameloblast with ruffled border. So when ameloblast is having a smooth end, it is less active and it absorbs enamel proteins. When the ameloblast is having a ruffled end, it is more active and it has lot of lysosomes, and that will cause endocytosis of enamel organic matrix, and it secretes calcium for mineralization. Also it secretes bicarbonate to maintain the pH. So the process by which the ameloblast alternate between the smooth end and the ruffled end is known as modulation, and it occurs in every 6 to 8 hours. Next is protective stage. After complete formation and mineralization of enamel, ameloblasts secrete a basal lamina-like material in between the distal end of the cell and enamel, that is called hemidesmosomes, and that will help in the firm attachment of ameloblast to the enamel surface and also to establish the dentino-gingival junction. In this stage, the main event is the formation of the reduced enamel epithelium, that is, the outer enamel epithelium, stellate reticulum, stratum intermedium, everything collapses and form the reduced enamel epithelium. And the function of reduced enamel epithelium is the protection of the newly formed enamel. It separates the newly formed enamel from the surrounding connective tissue. When the enamel is in contact with the connective tissue, the resorption of enamel can occur and the cement may be deposited over the enamel. So, the function of reduced enamel epithelium is the protection of newly formed enamel. Next is desmolytic stage. Here you can see the reduced enamel epithelium covers the newly formed enamel, and they secrete enzyme to the surrounding connective tissue, and that will cause atrophy to the connective tissue, and this will aid in the formation of pathway of eruption. Premature loss of reduced enamel epithelium that will prevent eruption. So let's summarize. Morphogenic stage. This stage occurs in early Bell stage, and in this stage the cell is low cuboidal or short columnar cell with large nucleus, mitochondria evenly distributed, centrioles and Golgi bodies at the proximal end, and the cell rests on the basement membrane. Morphogenic stage determines the morphology of the tooth, hence the name morphogenic stage. Next is organizing stage. In this stage, the inner enamel epithelial cell organizes to form the ameloblast, and it occurs in the advanced Bell stage. In this stage, nucleus moves to the proximal end and the mitochondria moves to the proximal end, centrioles and Golgi bodies to the distal end. The cell rests on the basement membrane, and this ameloblast induces the formation of odontoblast, and the dentin is secreted in this stage. Next is formative stage, that occurs in advanced Bell stage. Here there is formation of Tom's process, and this process is separated from the cell proper by the distal terminal bars, and there is formation of enamel. And here there is no basement membrane. Next is maturative stage. In this stage, the main process is the modulation. That means the ameloblast that alternate between the ruffled border and the smooth end, and this occurs in every 6 to 8 hours. And here the basement membrane reappears. Next is protective stage. In this protective stage, the size of the cell decreases and it becomes almost low-cuboidal, and basement membrane is still there. Here the main event is the formation of reduced enamel epithelium that protects the newly formed enamel from the connective tissue. Then the last stage, the desmolytic stage. In that desmolytic stage, the reduced enamel epithelium that will secrete enzymes to the surrounding connective tissue and cause the desmolysis of the connective tissue and aid in the process of eruption.